Good morning. Of course, we can't start until Nancy seats because, it's her show. And she's not hearing any of this. Hi, Nancy. Good morning, everybody. I'm Mark Roithmeier. I have the privilege and honor of serving as the CEO of the Alzheimer's Drug Discovery Foundation. Welcome to the 9th annual Melvin R. Goods Prize for Excellence in Alzheimer's Drug Development. As many of you heard me say before, and you'll hear me say this many times today, we at the ADDF, Howard, that's us, we at the ADDF, consider the Goods Prize the Nobel Prize of Alzheimer's clinical trials. And there's a little bit of a backstory to this. Let me start with Mel Goods, Nancy and her family's beloved Mel Goods, former CEO and chair of Warner Lambert in the 1990s. Led an incredible turnaround of that company. Any of you have ever heard of Lipitor? That was Mel. He also, during his tenure, brought the first Alzheimer's drug treatment to market. Ironically, over a decade ago, fell ill to Alzheimer's. He is still with us. He actually announced the idea of this award some 10 years ago, and what you can go and see on YouTube is one of the most poignant moments of film that you'll see in ADDF's history. His glowing spirit is with us, the twinkle in his eye, and Nancy tells us, and I think we've all experienced, he can carry a good tune also from time to time. So we're in honor of Mel Goods today. Nancy, your children, your grandchildren, your in-laws, your friends, thank you for providing this for us. Yes, Nancy, I know. Why do we have to have this at UN Week every year? But we'll get to that also. We are so fortunate for the Goods Prize this year. We have 5 of our total of 9 honorees through the years. Doctors Brinton, Orr, Rook, Waterstein, and at the end is Rhoda O, who is our awardee this year. You'll be hearing lots about her before the day's over. I do want to ask everybody to give it up for Marty, who's in the middle, who's the lone boy on the panel, showing that boy. Yeah, there we go. Very good. And I just want to say, and I think the panelists will agree, boys like science too, right? Word about Alzheimer's. You might have heard about it. It's been in the news lately. This summer, we've seen our first cracks in the walls of treatments that provide cognitive improvement. You'll hear a lot about that today. I like to think of it that up until this summer, Alzheimer's was a societal problem. It hit your family, you and your family alone had to figure out so much and what to do. But this summer, we're now transitioning to a medical problem. We can start to treat Alzheimer's. Let that sink in a little bit because you're going to be hearing a lot more about that today. What I'll tell you is that the scientists that are before you today, they, right here, right now, will be delivering the new biomarkers, diagnostics, preventions, and treatments. So this really is a special day. It's a special day every year, Nancy, but with all the news of the last year, it takes on even added significance. At this point, I'm going to turn it over to Howard. And I always love to do my introductions of Howard. Yes, Howard, I have a lot of slides, and I have video this time, so we good. Physician, scientist, lifetime of work in Alzheimer's. I want to tell a story from this summer. Myself and a lot of the scientists from ADDF were sitting in the back rows. We were in Amsterdam for a big scientific conference called AAIC. And this is where Eli Lilly announced the results of their new anti-amyloid drug, donanemab. And Howard had a family emergency, so he was streaming back from New York watching this. And they announced, and you're going to hear a lot about this drug and others today, that, look, it really looks very modestly, if not moderately effective in treating and improving cognitive impairment with Alzheimer's. Once it was over, I went up to the front of the room where all the Lilly executives were. And there are 2 leading doctors, Dr. Dan Scrivonsky and Dr. Mark Minton. And they both grabbed me, and they looked me in the eye, and they knew Howard wasn't there, and they told me the following. Now, this is after they announced the results of their drug. They said, make sure you tell Howard he was right. 20 to 25 years ago, he had this crazy idea for a PET scan. And that the PET scan decided who got into these trials, who didn't get into these trials, and were the trials effective. He said, and he got it right. He got it right on amyloid is not the be-all, end-all, but it's part of an answer. and he's gotten it right on PET scans, blood tests, biomarkers, combined therapies, the ultimate solve. So with that as an introduction, ladies and gentlemen, I give you for the rest of the morning, Dr. Howard Philp.
Thanks so much, Mark, and I just want to thank everyone for coming here today. I think we have an exciting panel of Goods Prize winners who I'll be introducing in a couple of minutes, and 5 other Good Prize winners who can't be with us today. But it's really become a terrific program over the years. And next year, the plan is for us to be in Stockholm. I hope I can announce that, or I'm not supposed to say that. All right, I didn't say it. Okay. The work that you're going to hear about today, none of it is about this amyloid that was approved over the summer, although the beta amyloid treatments are effective and relatively safe with proper monitoring, and really represent the first breakthrough in our field in history of having disease-modifying drugs. But I will say that they only slow it down by about 30% in general, and we want to slow it down to 100% if we can, or even prevent it. And I think the way that we're going to get there is through the novel approaches to therapy that are non-amyloid approaches that you're going to hear about today. About 10 years ago, maybe even 5 years ago, about 75% of the drugs that were in drug development were anti-amyloid drugs. This was a leading hypothesis for the last 40 years, since 1984, when beta amyloid was discovered to be in the plaques of the brain. But this year, this past year, 75% of the drugs in development are non-amyloid drugs targeting novel pathways like inflammation and genetic pathways, epigenetic pathways, which we'll go into today. And so this is what we envision as the future, which will be combination therapy. It'll look something like cancer, where we'll have multiple biomarkers and blood tests, as Mark mentioned, and digital tests, which we're going to hear about today, that will subset patients into their own buckets, like in cancer, and then have targeted therapies. People will be on 3, 4, 5 drugs in the future, I believe, to really get a hold of stopping this terrible disease. I particularly want to add my thanks to Nancy Goods and her family, and Mel Goods, for supporting this tremendous award and being with us for so many years at the foundation. It's really been a wonderful relationship that we really appreciate. Let's see. I think that's pretty much my introduction here. Let me get right to our panel. Our first panelist is Dr. Roberta Diaz-Brinton. She's a good friend. Robbie and I actually sat down on rounds at the Rockefeller University in 1981, which was probably before most of you were born. And we were with our mentor, Dr. Bruce McEwen, who was considered the father of what's called the neurobiology of gonadal hormones. In lay terms, it's really the effect of estrogens and testosterone on the brain, the impact of the menopause on the brain, and this has been Robbie's life's work, and we remain very interested in this whole area of the menopause as a risk factor for women, which might account for why women are at greater risk for Alzheimer's disease. So Robbie's an old friend. She's the inaugural director of the Center for Innovation in Brain Science at the University of Arizona, where she also serves as a professor of pharmacology and neurology. And she's going to tell us a little bit about her phase 2 clinical trial of a compound she's been working on for a very long time.
Yeah, a very long time. Thank you, Howard. And this is one of many innovative ideas that ADDF has supported and consistently supported over many years. Nancy, I want you to know that I have Mel's photo right next to my desk to remind me of you and the Goods Prize contributions. So I'm thrilled to announce 4 things. First, we're recruiting for our allopregnanolone phase 2 clinical trial to regenerate the Alzheimer's brain. So we're very excited about this, and our first 2 clinical trial sites are up and running, and we will have multiple sites across the country. So stay tuned for more updates. The other is an update from our previous clinical trial of allopregnanolone in persons with Alzheimer's disease. And this is the outcome of an ADDF-supported project to Dawn Matthews to develop a strategy whereby you can take the MRI and analyze that MRI and determine whether that person who's been diagnosed with probable Alzheimer's disease really has the disease. And we applied Dawn's new strategy, which was just recently approved by the FDA and supported by ADDF for many years in development. And outcomes of that showed that our selection of persons to be in our first phase 2A clinical trial was right on target. All but 2 actually had that Alzheimer's profile. And of the people treated with allopregnanolone, over 60% showed an increase, a regeneration of their brains in just 3 months. So we're very excited about now taking this ADDF-supported strategy, Core Insights, and now applying that to our phase 2 clinical trial so that everyone that we're enrolling in the trial actually meets that Alzheimer's brain profile. So we're very excited about that. And for Howard mentioned our work on trying to understand why women are at greater risk for developing Alzheimer's disease. We have now 2 phase 2 clinical trials ongoing, which we're very excited about, focused on women and that menopausal transition and providing an intervention that promotes brain health and breast health. So now women don't have to choose between their breast and the brain. They can have brain and breast health. So we're very excited about that. And lastly, but far from least, is we're developing a patentable formulation of allopregnanolone and have started that work now to develop an intranasal patent formulation. And Carolyn will be also working on the microneedles too. Thank you.
Thanks, Robbie. I just wondered if you could step back for a minute and tell us a little bit about what allo is and how it might be related as a neurosteroid, as we call them, to estrogen itself.
Yeah, so allopregnanolone, how many women have given birth in the room? Okay. So you made a lot of allopregnanolone, or allo for short, A-L-L-O. You made a lot of allopregnanolone during the 3rd trimester of pregnancy. We've all made it. It's a natural molecule. And interestingly, during the 3rd trimester of pregnancy, the brain undergoes a 4-fold increase in size. It's generating that brain during the 3rd trimester of pregnancy. And we discovered, I laugh because this goes way back, Howard knows. We discovered that allo could promote the generation of new nerve cells in the brain, in the adult brain. And that led to many, many years of research and with Howard's direction to me, which was just cut to the chase, Robbie. Does it work? And in fact, it does. So, and one of the great things about allo is it's safe. It's undergone the most challenging safety test there is. It's safe in women of reproductive age with fetus, right? So it is very safe. Now we just have to make it patentable.
Thanks, Robbie. Our next prize winner over the years is Marty Waterson. And Marty's been investigating a very important pathway in Alzheimer's disease, which has been unrecognized for many years, which is inflammation. So the brain of patients with Alzheimer's, and Alzheimer's recognized this in 1906 when he first described the disease. He described inflammatory cells around the plaques of patients with Alzheimer's disease. And So, because of the focus on the amyloid plaques, there was just a lot of disbelief that inflammation played a role in Alzheimer's. It just wasn't thought of as an inflammatory disease. But there were some really interesting studies that showed that patients could have plaques in their brains, but if there was no inflammatory response about the plaques in the brain, there would be no progression of the illness. And that led us to try to fund some clinical trials of anti-inflammatories for the treatment of Alzheimer's. I actually was involved at Mount Sinai back in the 1990s, I guess it was, of a clinical trial of prednisone for Alzheimer's disease. And the non-steroidals have been tried, and none of them, at least so far, seem to have worked. But Marty's, through a biotech company and a long history that he's had in developing drugs, has been working on very novel anti-inflammatories that would be specific for Alzheimer's disease. And he's currently the John Searle Professor of Molecular Biology and Biochemistry and a professor of pharmacology at Northwestern's Feinberg School of Medicine, and the founder of a biotech company focused on inflammation in Alzheimer's called Neurokine.
Thank you. I must give due to Howard. Back in the 1990s, he had a little note out about specifically a class of inflammatory molecules called cytokines. And that is sort of what we focused on. And actually, in terms of NIH funding and acceptance by industry, neuroinflammation was considered naive. And the ADDF, we must pay respect to them for the seed funding and allowing us to get the preliminary data to bring in millions of dollars now in NIH funding, okay? And we were just, some of us were talking about how at the talks, the whole thing of biomarkers progressing during the disease, neuroinflammation was always way out here. People didn't know what to do with it. As things progress, it moved over to where it was coincident. Now it's actually occurring as a co-indicator of true disease progression. From the patient perspective, it is critical. It's part of the multi-shots we have to take on a complex disease. It helped, actually a positive for COVID, everybody now knows what a cytokine storm is. So to some extent, one class of our drugs we have in phase 2 trials explicitly address the cytokine storm. In the MedChem laboratory, in the pharmacology laboratory, the go, no-go decisions were based on, do we have attenuation in vivo of the cytokine storm? Sometimes that storm in progressive disease is very, very shallow. And our driving force in its early days were the statins. The idea was we wanted this to be preventive. It has to be very safe, taken daily with high safety, and the idea you would alter the progression. We don't call it disease prevention because at the time the disease was not well defined, but the progression. And so I can update you the fact that we have two drugs that are in the same class that affect this cytokine progression. They are in other neurological disorders. One is in a multi-center trial for hemorrhagic stroke at early stages. Another one is in a trial supported by NCI, a proof of concept about the Alzheimer's type dementia that happens with glioma and other brain tumor treatments. So people might be saved from the tumor, but their cognitive function and lifestyle is dramatically altered. So we're going to get insight across these multiple diseases about exactly what combinations will work. Key to this is drug-drug interactions. So we've heard about amyloid and the fact that you're going to have something complementing that. One of our third areas is to look for targets that complement that. And in one case, we actually have preclinical data in which we could take an Alzheimer's model in which they have the microbleeds, and treatment with the compounds actually attenuates those microbleeds. So it actually is not only safe in terms of the usual FDA designations of safe, but it also complements amyloid approaches. And the third one is one in which is a New York-based Neurokine. Is actually at phase 2. We already have the clinical drug manufactured at the kilogram level. They are out trying to raise funds and recruiting patients in different types of dementia, AD-related disorders. And that's in a target family called kinases, protein kinases. So That's a big deal in drug discovery. Kinases were first introduced as FDA-approved drugs in oncology. In the early 1990s, they revolutionized oncology. They were considered toxic targets for neuro. Our work, along with that of several other laboratories, have gone to extreme to show that in animal models and in FDA phase 1 criteria, healthy people being tested, they are not toxic targets. So we hope to be able to give progress within the next 2 years of moving into that target area. All of these are focused on neuroinflammation, a particular type of it that has these cytokines, these small toxic molecules that are yin-yang in life, homeostasis. They're important. When they get out of control, either chronically or the result of a profound event like brain injury or infection, they become what is called a cytokine storm. And so controlling that and also enhancing the neuronal effect, that's how we attenuate the damage they do, the neurodegeneration, as well as enhance the comorbidity of prevention, which is a lifestyle issue.
Thanks, Marty. Okay, I'm going to move on, if you don't mind, to the next winner, former winner. Dr. Jerry Rook is a research associate professor at the Department of Pharmacology at Vanderbilt University in the Vanderbilt Center for Neuroscience Drug Discovery. And she's working on what are called symptomatic agents. So much of what we'll talk about today in terms of drugs is drugs that modify the course of the disease. The drugs that are coming out from industry today, anti-amyloid, modify the course of the disease. But since about 1993, the only drugs on the market were symptomatic agents. And we really haven't had a novel symptomatic agent come to market since about 2003, I would say. So Jerry's looking at a very novel pathway involved in the illness, which is also going to hopefully add on to the symptomatic therapy that we can offer patients.
Yeah.
So tell us about that.
Yeah, thanks, Howard. Yeah, I think Howard made an excellent point in the beginning in that I think we all understand that there's not going to be this silver bullet, that there's not going to be one drug or one target that can cure Alzheimer's disease in every patient. I think it comes down to personalized medicine, and that another point is that that we've all touched on is like there can be patients that have this huge amyloid burden in their brain, but if they don't have inflammation associated with it, they don't really have any disease pathology. Or there are patients that have very low amyloid, but they have severe Alzheimer's pathology. And so I think in the end, it's going to come down to personalizing treatments for each individual patient. And you know, part of that is treating the symptoms. There are going, and I'm very hopeful for all of our endeavors to make it to the market and to actually have an effect on the Alzheimer's patient population. But there's going to be a large portion of the Alzheimer's patient population that are extremely advanced in their disease state and have severe cognitive deficits. And so our program is heavily focused on enhancing that cognitive dysfunction that's associated with mild, moderate, advanced Alzheimer's disease. And I have to, you know, give a shout out to Howard, because when we were in our program, you know, you get to a stage, and we were relatively advanced. We had declared what we thought was going to be our clinical candidate, and we needed to go through what we all call this dreaded part of drug discovery that's called the Valley of Death, and no one wants to fund it. So it's a series of safety and toxicology studies that the government doesn't like to fund because it's, you know, not something novel or new, but you've got to get through that in order to get into clinical trials. And that's when Howard and I started talking. This was, what, 10, 15 years ago? 10 years ago, I think about. And he was all for it, you know, and it really, truly helped catapult us, you know, through that stage. He also got us in contact with the Harrington Discovery Institute, which also helped us. It's a fantastic partnership that he has created with ADDF. And so we were able to get through that phase and actually partner our program with a pharmaceutical company, which has helped drastically, you know, navigate us through this process. But our hope is that, you know, we're targeting a very specific pathway in the brain. I think many of you, if you've had family members that have Alzheimer's and have been treated, the cholinesterase inhibitors, for instance, Nepozil, it's along the lines of that theory, and that unfortunately for those, the chemical in the brain that's diminished in Alzheimer's disease, it just floods your body with it, and it targets parts of the body that are not important for Alzheimer's disease in the brain. So there's all these horrible adverse side effects that's associated with it. And I really truly believe that's a lot of the reason that it's not that effective. So we've isolated, the one part that chemical in the brain is important for learning and memory, and we're specifically targeting that part of the brain. And our drug is meant to enhance the function of that target and then therefore enhance cognition in these patients. So I think we're getting there. We're working on it. We're working on an additional backup program, of course. You never want to just have one shot on goal. And everything is going really well, and I think that's greatly attributed to the ADDF and just how much of a visionary that Howard is. He's always forward thinking, he doesn't follow the herd, and he's always thinking outside of the box, and I think that's what's given all of us a great opportunity to advance our programs. So.
Well, thanks, Jerry, and everyone else that's complimenting me. I just want to say flattery will get you everywhere.
But it's true. It's not just...
But I also want to compliment my staff. I mean, I have I think 12 or 13 neuroscientists on staff now.
They're brilliant.
Back in the old days, it was just me, but now we have a great team of people that do all sorts of things. And Jerry, as an example of our venture philanthropy model, we've written about 150 contracts with biotechs over the 25 years that we've been in business, where we get returns, based on convertible notes and other financial instruments. But in academia, we look for a share of the royalties on licensing agreements.
Yep.
And in your case, with the Acadia Pharmaceuticals, I think that was the
Yes.
relevant program. I think it was like a $515 million deal, if I'm not mistaken.
Yes.
And so I just want to talk briefly about how the work that you did in basic science and the cholinergic mechanisms, real briefly, I don't know, but then that translated into a very large licensing deal from Vanderbilt to Acadia to further develop the drug with the heavy lifting that later phase clinical trials require.
It's very true. So you can do years of basic science research and you can prove or validate the target, which we did for decades, but there becomes an inflection point, which I like to call it the inflection point. You've got to get to a certain place to where your program looks lucrative in order to partner it. And that's, I think, where the ADDF came in, because even though we just had not de-risked it as yet. And once you get through those safety studies and you can prove, okay, it doesn't have any horrible adverse effects, it becomes an extremely lucrative program to pharmaceutical companies. And then they will license it for a significant amount. Of course, the further you get by yourself, the larger the project is, or the bigger the dollar amount that's assigned to it when you license it. And unfortunately, and we all know this, drug discovery is extremely expensive, and you need these kind of licensing deals in order to push it forward. And so that's where we got with Acadia, fortunately.
And just to say, we also shared in a small way with some of the returns that Vanderbilt got on that licensing deal.
Yes.
Let me turn to Miranda Orr now. Miranda is an assistant professor of gerontology and geriatric medicine, close to my heart, at the Wake Forest University School of Medicine. And she's really, we talk a lot about the biology of aging as our overriding strategy for how we approach drug development for Alzheimer's disease. But I think Miranda is an example of an actual gerontologist, who, not a neurologist, but a gerontologist who's taking ideas that she's learned in her training about aging, basically, and translated that into a drug therapy program for Alzheimer's. And the rationale behind that really is that aging, by far, is the leading risk factor for Alzheimer's disease. And so the foundation is very much on this promotion and adoption of translating this biology of aging knowledge, which has been around going on for 100 years, into new therapeutics. And Miranda's program is looking, I'm sure you'll explain this, at what are called senescent cells. So we all have aging cells in our body and our brains that won't die and become very inflammatory. And no one's ever tried to treat this in Alzheimer's disease. So her senolytic approach is very unique, and hopefully it's going to work.
Yeah, thank you so much, Howard. I love hearing Howard talk about my research. I actually think he does as good or better job than me introducing it. So I'm a translational neuroscientist, and what that means is I spend an equal amount of time in the lab as a scientist, what you might stereotypically think of a scientist, discovering things, testing them in mice, and an equal amount of time really thinking about clinical trials and executing early stage clinical trials. And the ADDF is supporting me through that entire translational pipeline. And my lab, so what our research is focused on is really understanding what it is about our cells in our body as we get older increases the risk of developing Alzheimer's disease. And we, the collective we in the field, have really identified some fundamental aspects of this process and recently have identified how we can target them. And so as Howard said, my lab is really focused on one aspect of aging called cellular senescence. And these are really interesting from a biological standpoint and also interesting to the general public because they've earned this name toxic zombies. And so they make very good headlines targeting toxic zombies and Alzheimer's disease. And the reason why they've earned this name is because these cells are damaged. They should die. When you look at them under the microscope, they should be dead. They're carrying all of this damage, but they figure out clever machinery to survive. So you might think that that's a good thing. Your cells get to survive, but it turns out they're very, very bad neighbors. They release things from inside of them that kill healthy neighboring cells or turn healthy cells into senescent cells, and they keep perpetuating this chronic disease. And they contribute to all sorts of diseases of aging, and our lab found that they really contribute to Alzheimer's disease as well. And a really small percentage of them, we determined about 2% of brain cells in people with Alzheimer's disease are senescent, really do have a huge influence on tissue dysfunction. And so we tested whether or not if we clear these cells, so in Alzheimer's disease, actually killing cells has an improvement. And we found that in Alzheimer's mouse models, we stopped the disease from progressing. And these mice were equivalent to a 70-year-old person with advanced Alzheimer's disease. And at the point those mice started getting the drug, the disease did not get worse. And so with the help of ADDF, we have moved this to clinical testing. Just earlier this month, we reported results from our phase 1 trial, so showing that the drug entered the brain, that it was safe. And so now ADDF is supporting a phase 2 trial with a larger number of participants enrolled, as well as a placebo arm. And we are even starting to recruit internationally. We're working on that right now. And what the Goods Prize has done is really revolutionized my lab from the basic science standpoint. And this is so exciting to me. So up until very recently, if you looked at a tissue under a microscope, you could maybe look at 4 characteristics of what's happening. But now, with technological advances, we can look at hundreds or thousands, up to 18,000 features in each cell in a brain at the same time. And so the Goods Prize is allowing me to use this technology to look at brains from people that passed away with Alzheimer's disease and really understand why one senescent cell, this toxic zombie, looks very different than its healthy neighboring cell. And this is important because now we can identify new drug targets, test them in mice, and what we can do with this technology from mouse brains is compare treatments, see which ones are more effective, and then move back to the clinic. And so my lab is really using this iterative approach from discovery, moving to early stage clinical trial.
Well, thanks, Miranda. It really is novel, and I think we don't really know any other programs like yours in the world. Maybe Dr. Kirkland is about the only one I think we know about. But it's really so promising. I'm going to move to Dr. Orr, if you don't mind. So finally, I'd like to introduce our Goods Prize winner for this year, Dr. Rhoda Orr. Dr. Orr is a world-recognized leading expert in the use of digital technologies for applications in disease, and particularly Alzheimer's disease. She works with the very famous longitudinal study on aging called the Framingham study, and has been working to create digital biomarkers to detect the disease very early, to assess patients on cognition and function and other aspects of the disease that can be detected on your smartphone. So this is going to be a tremendous advance in the way we do clinical trials and the way we assess people in clinical practice, to have digital biomarkers instead of just having interviews and paper and pencil type of things, being able to do this in a really expedited way. It'd save a lot of money, I think, in terms of how we actually conduct trials. She's currently a professor of anatomy and neurobiology at the Boston University Chobanian and Avedissian, did I say that right? School of Medicine. And I guess ADDF started funding her about 5 years ago. And we're working together with Rhoda in the Diagnostics Accelerator, which is our $100 million program to develop diagnostic tests and better biomarkers for Alzheimer's disease with Gates Ventures and a number of other donors. So let me turn it over to you, Rhoda, to give your remarks.
Great, thank you. So, as the newbie on the bench here, I was really impressed with how amazing work that they've done and how far they've advanced things. So I'm kind of crossing my fingers that years from now I'll be where you guys are in terms of how much you've moved things forward. But as Howard said, you know, I've been on this mission. I'm a neuropsychologist, so I've been on this mission about how do we assess cognition a lot better than we have. So, you know, traditionally we've done it through these paper and pencil tests. And when I first started years ago, you know, we were still thinking about Alzheimer's disease really in the symptomatic, really at the moderate stages. And then over the years, we've gotten better and better at sort of identifying this disease earlier and earlier. So we've moved it into the mild, and now we're at the MCI, and now we're trying to even move further. So that's made my challenge, actually, in trying to figure out how do I detect those earliest signs, you know, which is the cognitive and behavioral related symptoms even better, right? And the other thing that we're now finding with these new treatments is now there's hope. There's like real hope, because if we can do it early enough, we can actually, now we're starting to see, we can maybe slow progression, right? And then the next goal is, well, if we do it even earlier, can we stop progression? And then eventually, if we do it early enough, can we prevent it altogether, right? So that's sort of the importance, at least I see, of trying to figure out how do we find these cognitive, you know, assessment tools to allow us to do it. And as a neuropsychologist, you know, when you give a test, when you give these neuropsych tests, these traditional paper and pencil, it turns out that when people are responding, they give you lots of responses. They give you responses that lead to the correct response, but it turns out not all correct responses are exactly the same. And then they'll also give you incorrect responses. And it turns out not all incorrect responses are always the same. Sometimes, you know, a correct response is a little less correct because it took them a while to get there. And it's, on the same hand, an incorrect response, it was kind of close, right? But it wasn't quite there. And we lose those nuances when we do our paper and pencil tests. So the question is, how do we capture that richness of these responses as people are responding that we aren't, you know, hadn't been currently? And so that's where the concept of digital, when digital came on the scene, it became suddenly, wow, we now have a technological tool that will allow us to pick up much more subtle responses on our way to whatever that final response is, right? And so, as Howard mentioned, you know, I've sort of gotten tagged a little bit as being, you know, pushing forward the concept of digital biomarkers, because that's really kind of what got into my head, is what happens now is through these digital tools, we can start to identify these very accurate, meaningful changes at extremely subtle periods, right? better than we've ever been able to do. I do want to mention, though, when I first started to put those words together, digital and biomarker, I actually got a lot of pushback. I got people telling me, you can't use those two words together. They don't mean the same thing. And it was the ADDF, where when they heard me put those two words together, they actually not only didn't poo-poo it, they actually embraced it. And so consequently, through the ADDF, I've been able to push forward this concept that I've had for a while, but really bring it into reality. So what have we've been able to do? One of the things we've really honed in on is the concept of the voice, digital voice. So why digital voice? Well, it turns out that speaking is a really cognitively complex task. So when you're speaking, you're actually reflecting lots of different cognitive processes. And the question is then, how can we capture those? How can we measure that? Then on top of that, it turns out most people speak. So suddenly we have a way in which to maybe we can assess people's cognition, no matter who they are, no matter where they are, no matter what their language, what their education, what their culture is, right? And now, because of the smartphone, suddenly we have a very powerful tool in which to collect this digital voice. It's so easy to do. And that's really what the Diagnostic Accelerator did, is they were really the first now to step forward and understand sort of at least what was my strategic vision at that time and say, yes, this is a means in which we can start to think about how do we assess cognition in a way that allows us to be maximally inclusive. Because up to this time, because our cognitive tests were such, they were very biased. They were biased educationally, right? They were biased culturally. A lot of them were developed within the Western culture. So it was very hard to push forward from a global standpoint. And now suddenly we had a mechanism in which maybe we can embrace the whole world, which is undergoing this global epidemic. So, so I want to give you a little preview, because we've now, thanks to our initial funding through the ADDF, we've now been able to collect enough digital voice recordings, and we've been able to expand it and to collect it from other languages, cultures. So what we've now been able to do is we've taken voice recordings that were in US English, Malay English, Mandarin, and Cantonese, and we're able to analyze the acoustic features of these digital voice recordings and find that there are common features across all of them. We've now started to collect also, we have digital recordings in Swahili, Hindi, Tamil, and so our next step is actually now to combine across these multiple languages. And what we're trying to get to is the first attempt to really do a cognitive assessment that's going to be globally possible. And so that's where we're at.
Thanks, Rhoda. And you know, I just imagine, like in my small clinical practice, I do paper and pencil. And one of the things I do, for example, is a clock drawing test, which is actually quite hard for people with cognitive impairment, and tests multiple domains. But I know that you were involved in the commercialization of a digital clock test that because it's done on an iPad or some device like that, it collects so much more data than just asking somebody to draw a clock, and it's a great example. So I really look forward to the day in my practice where, you know, I'll be able to capture somebody's speech. And, you know, we see this on TV, I think, as we went through the old Reagan presidency, his speech, people were questioning his speech because it was the earliest sign of his cognitive impairment. And we all have word-finding difficulties as we age, and naming and remember people's names. So that's kind of also reflective, as you said, of how speech reflects cognition and is very sensitive because it's so complex. So you've made great advances. One of the things about our venture philanthropy model is commercialization. And we don't want to see this work just sit on be part of a paper that gets published in a medical journal or somehow stays in academia. And so could you talk to us a little bit about how the digital technology, and maybe even add in a comment on artificial intelligence, how this digital technology is being rolled out commercially, what you think was going to happen in the coming 3 to 5 years with digital technology for the assessment, and also, of course, in the use of, for example, speech or other digital technologies in clinical trials, where it's really changing the way we do clinical trials. I know, I'll just say one more thing. I know in my practice, if I refer someone to a neuropsychologist, usually they're undergoing anywhere from 3 to 6 hours of testing, and I get these huge reports, and I'd rather just say, okay, speak into my phone, thank you very much, and bill them $5 or something like that.
Right.
So maybe you could tell us about the commercialization issue.
Yeah, I think you hit on exactly like the importance of commercialization, because when I first started training in neuropsychology, that was the same thing. I was taught that I have to give this test protocol that literally lasted a day. Like, I would come in, test a person, and my only thing for the entire day was to test one patient, right? And then to write up this very, very lengthy report. And I mentioned to you earlier that when you're giving these responses, you're giving lots of responses. So I'm like trying to capture all these little responses, and it's taking me an entire day to do it. And the reality is, it's great from a research standpoint that I can sit there and follow, one person at a time, for however long, and score everything, but it's not feasible when you want to try to bring it to scale. So if we talk about the digital clock drawing test, we used to score the clock by hand in order to get to these objective measures, and it would take us 20 to 30 minutes to score one clock, right? When my colleagues at MIT and my neuropsychology colleagues as well, David Laban and Rod Swenson, when they actually went to create a digital version, they had no interest in commercializing it. I just want to let you know. So I brought it in, and we used the digital version, and I thought it was amazing. We got over 1,000 different metrics now. I used to hand score and get like 10, right? And now we get over 1,000 different ones, but you know what? It was still taking a lot of time internally to process it. And I said, you know what? This is an amazing tool. We got to get it out there. There are clinicians all over who are using the clock drawing test, and yet here's this digital version, right, that could really make a much bigger difference. But we can't do it at the research level. And so I really pushed them to commercialize it. So now, which they did it did take me a long time, but they did finally commercialize it, and their commercial version, you draw the clock, you dock it, and boom, you get a read back. And that's what we need to get to, right? So that's the same thing now with the digital voice. We're going through and we're doing the research and the science to figure out, how do we need to analyze it? How do we use artificial intelligence? Because we want to take advantage of the three-dimensionality of that voice, right? It's got some audio features, it's got linguistics features. We can't be hand scoring it, we can't be deriving it. We need AI to take the complexity of that and analyze it and do it in an automatic way. So just like we now can do with the digital clock, you can give boom, a response. We need to be able to do that. And the only way you can do it is commercialization. Here's the problem when you do things on smartphones, when you try to do it as a researcher. Every time there's an upgrade, in the iOS operating system, in the Android, boom, everything stops working. So the one thing you don't want to do is you don't want to create something that every time there's an upgrade in technology, which is happening all the time, it stops working, right? And the only people who know how to do that is the commercial. They're the commercial entities. They're the ones, and that's what I decided. I decided I don't want to be in the business of worrying about every time someone does an upgrade in their phone. I want to be in the business of understanding what this means and working with the data scientists to figure it out. And I want the commercial companies to take this, run with it, push it out there, and make it global.
Thanks, Rhoda. I think maybe we'll turn to questions from the audience at this point. Yeah, so if anyone has any questions
We're going to have somebody come around with mics, so if you have questions, there's one group in the room Howard alluded to that I just want to point out. With the ADDF, we're about multiple shots on goal, right? And you hear across a variety of areas, and it's always been about three things: biomarkers/diagnostics, prevention, and therapies. And the 13 PhD scientists we have on staff are all in the back of the room, and they make it all happen with Howard. So thank you, guys. Great to have you here today, which is good. All right, first question. There we go.
Thank you. Thank you, Howard. Thank you, everybody, for all your efforts for all these years, for the pencil and the paper. And just my question is really, I was waiting for, no one was talking about AI until you mentioned it. And, you know, one of the thoughts is that we're at 30% success rate, 40% success rate. Where is everybody else in your studies looking towards the science of AI, where we feel we're going to destroy the world with, but how can we also save the world with the opportunity of asking for help from something that can work at maybe 100,000 million times faster than we can, in supporting some of this data that we already have as a benchmark, and saying, okay, let's run with destroying the zombie cells. Let's figure out a way for us to get there quicker, I guess would be my question.
Does anybody want to? I mean, I think what we've seen with artificial intelligence so far is really at the very earliest stages of drug discovery. There are what's called libraries out there. And these are basically masses of molecules, of compounds that exist, sometimes up to 1 million compounds. Like industry would have a library of multiple compounds that could be used for different targets. And so the first phase of drug discovery is often screening these libraries. And as I understand, I'm not an expert on this, but artificial intelligence has accelerated this process greatly, so that what used to take 6 months or a year or months to screen a library of 1 million compounds on your target in the laboratory to see if it actually has any potential for working. Now there's a way using artificial intelligence that we can accelerate that tremendously. And I'll turn it to my colleagues to,
I think, another thing is machine learning, of course, is very big right now. So the brain is extremely complex, and Alzheimer's disease is a perfect example of that. There's so many different pathways that intertwine. It's extremely difficult, like paper and pencil we've said before, to try to figure out which pathways are important, how they interact, how do they interject or intercede each other. And you can use machine learning to try to figure out which targets are really essential in these pathological changes and which targets we should try to pursue. And so I think it's a way of accelerating the drug discovery process of just finding new targets and new things to investigate. And so there's been a large push recently at all the levels, but like most of us, we review grants for the government, and there's a lot of machine learning grants out there that I think are extremely interesting and can really, really excel the field if we pursue them. So.
And one other component that I talked about earlier that ADDF supported and that now we're using is the algorithms that were developed through Don Matthews using artificial intelligence and looking at the complexity of these structural changes that happen in the brain, right? And they're happening all over the brain. And now developing, well, this is an Alzheimer's pattern. This is a Parkinson's pattern. This is a frontal temporal dementia pattern, right? Whereas before, these diagnoses were made by the clinician and with imaging with the input of imaging. But in this case, now we can actually have the images, the MRI images, pre-analyzed before they're enrolled into the trial to say, this person actually has that complex profile of the Alzheimer's degeneration in the brain. Oh, but this one has frontotemporal, so they're not really appropriate for your trial. These people are. And so, but essentially that's billions, trillions of data sets that then went through this machine learning, this artificial intelligence for learning what's an Alzheimer's profile of degeneration versus others. So I think it's, and part of what I was saying to Howard earlier, what I'm really hopeful for is that now having this degree of specificity, we can now actually accelerate the conduct of our trial, potentially not having to have, I know, hundreds of people in the trial, but having less, which means we can move faster and more accurately at the same time, which is a remarkable combination. You actually have accuracy and speed.
Yeah, I think it relates to my experience in practice. Even today, I'll order an MRI of the brain to see if a patient has strokes or if they have what's called hippocampal atrophy, which is the part of the brain that shrinks. And I'll get what's called a visual read. In other words, the radiologist will look at it and go, yeah, it looks pretty small on that hippocampus. I think I'm going to report it as hippocampal atrophy consistent with Alzheimer's disease. But I never order the MRI without asking for an analysis by a company called Cortex that has a product on the market called NeuroQuant. And NeuroQuant is already on the market using some of the principles that Robbie was talking about, where they do the MRI, and then they put this overlay of computerized analysis from the MRI, which is basically generating data. And then they'll give me a report that the hippocampus is at the 5th percentile for the person's age compared to a database of 2,000 people. Or the hippocampus is at the 90th percentile, and then I know there's no neurodegeneration. We funded Dawn Matthews' company a number of years ago to even take the next step to go forward and do it in an even more sophisticated way, getting much more sophisticated reports. And she just started out like kind of by herself as an entrepreneur, but she's become very successful with the product. And so I think we're starting to look at the next generation. And if you think about the application of this kind of computerized analysis in radiology, think about breast cancer, lung cancer, and how data analysis of those is generating much better interpretations of mammograms, for example, using technology to analyze the data. So I think that's a place where AI, and as Jerry mentioned, in the brain, it's so complex. And as Robbie mentioned, usually we're just talking about two regions, the frontal lobes and the hippocampus, but there's so much more going on. So it's really exciting to think about what the future of neuroradiology and neuroimaging is going to be. I would like to see the application of this artificial intelligence approach to the PET scans. Because right now, the PET scans are very subjective reads. So if I send somebody for an amyloid PET scan right now, I get a very subjective, it's usually like the brain's full with amyloid is kind of the response. So I think we're going to see a lot of it, or particularly in the Alzheimer's space, a lot of artificial intelligence, or whatever the right word is. I don't really know what artificial intelligence means. I don't know how you can be artificially intelligent, but any other? Yes.
Two separate questions. How comfortable do you feel about the ability to measure inflammation and degrees of inflammation? And the other one has to do with the digital work. How do you separate culture within one language in the data?
Well, I'll take a stab at the inflammation. We have biomarkers now in the blood and in the spinal fluid for inflammation. And so the next step is what's currently happening in clinical trials and in observational studies following patients, is that these biomarkers are being utilized. One of them is called YKL-40, another is GFAP, GFAP. The names don't mean anything, but the advance is really that we have blood biomarkers of neuroinflammation, and they're being developed, and I think they're going to be commercialized I would say in the next year or two. They'll be commercialized, and that's going to be a big advance. They're already being used in clinical trials, but I think when they come into the clinic, it's going to make a big difference. We're going to be able to start to subset patients. So we'll have patients that come in with cognitive impairment. Maybe they'll go for the amyloid scan. They'll be designated as Alzheimer's disease, and then we'll say, oh, maybe they have inflammation and they're going to progress, or maybe they won't. And if they have inflammation, maybe they'll go into a clinical trial of some of the companies that we're supporting for this neuroinflammation. So, and I have to tell a story. I trained as an immunologist over at Rockefeller in the 70s. And then when I started doing Alzheimer's, I realized that there was inflammation in the brain. So around 1984, must have been 85, I went down to the NIH and I said, you know, there's inflammation down there in the brain and we should really start to target it. And they basically kicked me off campus. You know, they just said, you know, basically you're you know, we're focused on amyloid. So what we've had is a real delay in the interest in inflammation, but it's come around. And I think part of it is the biology of aging. I mean, inflammation is one of the primary hallmarks of aging, but it's recognized, it's very well recognized that inflammation is playing a very important role in Alzheimer's. And I'm talking with Lilly and Pfizer people and other people like that, where there's very conservative approaches to new targets and new areas of interest. And they're saying, you know, we can't talk about it, but we're looking at inflammation now. So we've got the gamut from university scientists all the way through biotechs and into industry, you know, large pharma now on inflammation. So I think it's the next era of what's going to happen.
I can specifically address that in terms of the testing. There's a company in St. Louis that's really pushed the envelope forward, and the key thing here is precision and quantification, because you want to make sure that the samples are collected at your clinical sites, and they've worked closely with most of the academic scientists who are doing clinical trials and coming up with what's called a standard operating protocol, which they put forward, and that profoundly affects it. And they're coming up with what are called sets or profiles. So neuroinflammation comes in many different flavors. So they have an initial set that will tell the clinician or the trialist that what you have here fits probabilistically with a certain type of neuroinflammation. That actually helps for recruitment, because one of the big problems in recruitment in early phase trials is you want to have people that your drug works a certain way, and you want to make sure the people you're bringing in, at least a lot of them have that. So you can profile this. And the big challenge really is getting that refined over the past 5 years to get that high sensitivity and precision. And there are reports in the literature now that they have this, and there will be some coming out within the next 12 months. But that actually is a criteria that are used. And when we review proposals from academia and industry, as scientists and consultants, they all have this in one form or another. And the big debate is, you know, what type of neuroinflammation do you have? And for me personally, I'm glad they focused on the cytokine surge. There are 2 sets they have, so we can actually start measuring that clinically. And the idea is to fund that. We're actually, for our phase 2 trials, we're actually trying to put that into practice.
And I think I'd just comment that the company in St. Louis that you're referring to is C2N, if I'm not mistaken.
Yeah, it's one of them, the other.
So, well, I mean, I just should say that the first blood test on the market is from it's available in 49 states now. We're waiting for it to become approved in New York, which has the strongest regulatory environment for bringing tests to market and for medicine. But we were very early investors in this company C2N, which has the first blood tests for Alzheimer's on the market, coming out with a new one. So we do think that they'll be going into other areas going forward. And so, you know, I agree with Marty that, you know, it's already possible in 49 states that a person comes in to the doctor's office with a memory complaint, and imagine, you know, 5 years ago I said they could get a blood test and within, you know, a day or two have about 90% precision that they do or do not have Alzheimer's disease. That's changing the world. And we were early investors in that, as well as very early investors in the brain scan. And what's been shown in terms of how this blood test was brought to market is that the blood test was highly correlated with the brain scan, which was the kind of the golden standard. And so for the FDA to approve the test, the company had to show that the test actually correlated with the brain scan. Maybe a lot of people don't know about it, but this is the new marketplace for Alzheimer's disease. It's really, in my view, I've been doing this 40 years. If you asked me 5 years ago there would be a blood test for Alzheimer's, I'd be very skeptical, but now there is. Let me turn it over to Rhoda to answer your second question, if any of us can remember that one. It's been a while.
Yeah, no, I got it. So one of the things I would start with is just to remember everything we do through our brain. So anytime we do anything, we're actually always emitting our cognitive capabilities. It's just about how we capture it, right? So one of the things that we started with, for instance, with the digital voice, is we paid attention to what are the test stimuli. When we ask people to respond, how do we ask them respond? And we need to let them respond from their place, from where they're coming from, right? And I know that the DXA Speech Consortium team is in the back there, so they know how much we spend time worrying about how do we elicit that voice that we're, in a way that will allow anybody, independent of their culture, their education, their language, right, to elicit a response. So one of the things we did was we took the picture description tasks, which traditionally have been for many, many years the cookie theft picture. So it's this old picture. It shows this woman standing by the sink, with water overflowing, washing the dishes, and there's two children in the background, and one's on a stool, and it's kind of tipping over, and they're trying to reach for the cookie jar. I can't tell you how biased that is culturally, right? So what we did was we decided we need to change that picture, right? So we did. We hired actual artists from all over the world, and we asked them to come up with scenes that were much more common regardless of where you were in the world, right? And then we asked them, describe what you see. So they're describing it from their frame of reference, right? And then afterwards we asked them, do you remember what you described? So we're getting a memory test, but it's in their words, it's in their way, right? So that's how we get around this. It starts with the stimuli, and then it understands that when we do these natural behaviors, we're pulling out that cognitive behavior. Now we just need AI, right? We need the data scientists to come in and help us analyze now these very individualized responses in order to find where are those common features that hang across everybody, regardless of what language they started with. So I hope that answers your question.
Any other questions? I think while we're waiting, I didn't ask Miranda a question. Miranda, there's other, besides inflammation and senolytics and senescent cells, there's other pathways in the biology of aging. I wondered if you could speak to one or two of those. I was thinking in particular, we're starting to see programs in a process called autophagy, or the failure of autophagy, where it's been a very difficult target over the years to drug, as we say. But we're starting to see companies that have targets for autophagy to, I'm going to use some technical words here, increase lysosomal acidification, for example, and things like that. I wonder if you could put into everybody's, using words we can all understand, sort of from the biology of aging point of view, why is autophagy important? And particularly, let me just say that we know now that Alzheimer's disease is not just plaques and tangles. It's alpha-synuclein, Lewy bodies, TDP-43, tau, and these are all misfolded proteins. So we need a parsimonious approach to misfolded proteins because they're not being removed by the autophagic mechanism. And if you could comment on that.
Yeah, I spend a lot of time thinking about this, and so at high level, what Howard is talking about is as our cells are functioning, we make new things and we need to get rid of old things. And autophagy is part of this garbage disposal system where old things get degraded and removed. And there's a lot of evidence that with aging, this process goes awry. And so there's a lot of interest in targeting it because if we can keep everything, shiny and new, that seems like a good thing. The problem is that every cell in your body is not the same. Every tissue in your body is not the same. And increasing this process in some of your tissues is very bad, whereas increasing it in other tissues is very good. And so some of the early studies, just one treatment trying to globally increase it across all tissues in mice looks really good, but there's data from early phase clinical trials that seem like it's not so good for the brain. And so getting to tissue specificity. So not only we have this great drug target that we think is really important for the brain, how do we get it to just the brain so that you're not, damaging your skeletal muscle or your liver or other tissues? So it's a very complicated issue. And aging in general, where the field is moving towards is really understanding how each tissue ages differently and collectively. And so we're just really tip of the iceberg with that. And some of the technologies that I was mentioning that we're using in our lab is really helping to understand this. We're trying to understand the difference in cell age between cells that are right next to each other, because cells in your body are not aging the same way. And so really understanding that is going to help us develop smarter treatments.
And, we funded a scientist who was at Harvard at the time, and he's now at BU, named Tom Perls. And he was conducting something called the Centenarian Study. He had, I think, around 700 families around the world where everyone in the family in a generation lived to be 100. And the chances of that happening was like 1 in 10 trillion or something like that. And so he was studying these families. I remember he had a picture of all the family members on a couch at a reunion, and there was like the 110-year-old matriarch with the 90-year-old daughter, with the 70-year-old granddaughter, with the 50-year-old great grandson, with the 15-year-old, and there were 6 generations alive. And, he thought that he thought, why is this happening? And he came up with a genetic theory. Obviously, this is a genetic mutation, not a mutation, but a genetic phenotype that allows people to live to be 100 or 110. And it's clear that people that live to be that ripe old age have siblings that usually are too, and parents that usually are too. He was looking for one gene. He started a biotech company. Of course, it wasn't one gene. It's polygenomic, right? Why, with aging, some people get diabetes and other people get Alzheimer's. And so it's kind of interesting how genetics actually plays a role in aging in a way that we don't typically think about. And it's also interesting, last thing I'll say, is that in characterizing these people, he found that their menopause is delayed by 10, 15, 20 years. The onset of diabetes and hypertension is delayed by 10, 15, 20 years. What can we learn from the study of centenarians to develop drugs for Alzheimer's disease?
So I want to give a plug for, there's a series on Netflix called Blue Zones. And if you haven't seen it, I watched it and I thought it was done very well. And there's even, at the very last episode, talks about how to create blue zones. So blue zones are areas in the world where people live exceptionally long and healthy lives. And I'm not going to give anything away, but you will notice as you're looking at these blue zones that there are very common features, and it doesn't matter if it was Japan, the United States, or Italy. There are some very common features, and it's largely in how we live our lives. There definitely is a genetic component, but the very last episode talks about how can you make your community a blue zone, and there are now, you can apply for your city to become a blue zone city, and this is something that I'm really interested in for my city, and I think any city, because it really increases healthy living, so going into prevention, but it's I think really thought-provoking, and I think really hammers home the point that it is very complicated how you can live a long healthy life, and genetics and environment interact.
Okay, we have time for one more question from the audience. Is there one more anywhere? There we go, Marcia.
Resilience is what you're going to study.
Yes, thank you everyone for enormously creative and exciting experimental journeys that you are on. I have 2 questions, one for Dr. Diaz-Benton, and that's related to, I'm fascinated by the information that you presented on allo production. Is it known what stimulates the increase in allo production in the 3rd trimester of pregnancy? Are there any known behavioral correlates to these changes in size in terms of learning and memory? Are the changes preserved postpartum? And if in size and function, and if so, for how long are these changes preserved or are they lost? There was some research that went on back in the 70s, so predating 81, before people were born here, that was done at Purdue University looking at women postpartum. And the psychologist that was describing the behaviors described it as cognitive scatter that she identified that persisted up to 3 months postpartum. They had behaved, they had psychological correlates and analyses that had gone on all through pregnancy and then in the postpartum period. And my question for Dr. Al is, what are some of the acoustic parameters that you're examining in these responses? Thank you.
Okay, so you get an A for your bio exam. So allopregnenolone is a daughter of progesterone, and progesterone is in large quantities because it's maintaining the uterine environment, the uterine lining that will support the infant. And so allopregnenolone is a daughter of progesterone. It's made from progesterone or can be made directly. And you're making great quantities of that during the 3rd trimester of pregnancy. And something that was humbling and surprising to me is that the concentration that is effective in our Alzheimer's trial is exactly the same concentration that women make during the 3rd trimester of pregnancy. And that essentially what the brain is telling us is that I'm still renewable. I can still generate those new nerve cells. The other aspect of allo's action, which was surprising to me, was that allo can promote the energy system of the brain, the actual generation of energy and regulation of mitochondrial function that was talked about earlier. And it's anti-inflammatory. So it has multiple multiple points of impact, so that it's promoting regeneration, but also, very importantly, it's also looking around its environment and saying, oh, this brain needs more energy, and it promotes the mitochondria to generate that ATP energy. And it also reduces the inflammation. So part of what we see in these neurosteroids that Howard referred to earlier is that they're a systems biology regulator. So they regulate and promote whole systems of biology. They're not a single target, they have multiple outcomes. So I think in certain instances, that is actually going to be very effective, because if you're generating new nerve cells in the brain, you want to make sure that you're also reducing the inflammation in the brain that's going to harm those new nerve cells. You also want to make sure that there's sufficient energy in the brain now to incorporate those new nerve cells and those new nerve cells to work. Right? We also found that allo, again, didn't know this until we started doing MRIs of people's brains, allo promotes the integrity and the generation of new white matter in the cell, in the brain. Now that white matter are those tracks in the brain that you're using right now to whirl around in your brain in very fast synaptic transmission. It's like, oh, I've heard that before. Oh, neuroinflammation, Marty was just talking about that. Oh, mitochondria, Miranda was just talking about that. That's all white matter, right? That fast synaptic transmission. So what we see with allo, to your point, is that it's multiple systems of biology that are being promoted, right? And importantly, not being promoted in an unregulated way. It's very regulated, and that's what contributes to its safety profile. Did I answer your question?
Yeah, so for acoustic features, when you're speaking, there's sort of both the sounds when you're making them. So you'll have like different pitches, tones, right? You can imagine that, right? There's also when you're not speaking. So you have pauses, right, and different kinds of hesitations, a little bit of stuttering, things like that. So it's sort of pulling all of those acoustic features, because we all do that when we're talking, right? And you can tell when someone's surprised, when someone's angry, right? These are all modulations in our tone as we're speaking. But then there's also what I would call the non-speaking features, which are, you know, the elongated pauses, the stuttering that happens, you know, et cetera. So all of those combine create these acoustic features, which are a little bit different from obviously the linguistic features, which is really about your word choice, the way you form your sentences, et cetera. So right now we've been focusing a lot on the acoustic features. Our hope is actually to move into including linguistics features. It's a little bit more complicated when you go cross-language, but that is our second stage, just to let you know, because our research shows that acoustic features alone are pretty good at predicting people who are cognitively impaired versus not, and linguistics are also, but it's really the combined that gives you the most power. So that's where we're trying to get to. So I hope that answers your question. Thank you.
Okay, so stay seated. Lightning round. We're going to go quick, and Howard, don't worry, you get the last word. From the panel, we're going to start with Robbie. Quick. We talked about everything from the first drugs starting to crack the wall, the amyloid drugs. We talked everything about AI and how that's going to speed things up. Robbie, and we're going to go right down the line. What are you most excited about?
I'm very excited about the precision medicine approach, that there are going to be multiple therapeutics that actually are relevant to individuals, that now we can actually be able to say, this is what your brain needs. And I have to give Howard and ADDF and the Goods Prize tremendous credit for being visionaries in the field. Howard started ISOA, ADDF with multiple shots on goal, right? Knowing that we need a precision medicine approach to Alzheimer's disease.
All right, and Howard, don't worry, you get to agree or disagree with all of them. Marty, next.
I'll follow up with the same term, precision medicine, in terms of its application to neuroinflammation. We have already deconvoluted the sets of the types, the flavors of neuroinflammation, to where we know that the particular drug, even if it's precision in terms of the target, or whether it's broadly working like allopurinol, the type of drugs you're going to develop for each one are really critical for this being able to engage precision medicine. And we now have the biomarkers that are directly related. The sets, they're not one single ones. They're not the magic test. It's a set of diagnostics. And the thing I'm excited about is that will soon be a standard in clinical trials. And within 5 years, I think we'll see it at the clinical office.
Jerry?
I think we're probably all going to have a general theme, but along the lines of personalized medicine, what excites me the most, and we talked a little bit about it, is the machine learning and the AI and using these new technologies. As someone mentioned in the audience, like people are afraid we're going to ruin the world or destroy the world with it, but we can also use it in a positive way to help, you know, cure and build. And so I'm very excited about machine learning and the use of it to identify new targets to find cures for these diseases and Alzheimer's
Dr. Orr.
disease.
I agree with all of those, and I will add another level, which is, I think, actually, Dr. Brinton touched on it, combination therapy. So with all of these things, we are able to use combination therapy for precision medicine based on AI to determine which targets we should drug. So
Fabulous. Rhoda?
So I actually am about precision brain health. And I think that we're on the pathway where we're going to figure out how to optimize our brain across our lifespan. And I think that that's what's going to be really exciting, so that instead of just focusing on treatment, we're going to be on prevention.
Which is great. Howard, are they all crazy? Do you agree? Do you disagree? Last word.
Well, what I'm most excited about is I remember seeing my first patient with Alzheimer's disease in 1980, having graduated medical school in 1974, trained in New York, saw hundreds and hundreds of older people, never made a diagnosis of Alzheimer's, never heard of it. It wasn't in my textbooks. And then I walked into a facility and people said, these poor people that are bedridden have Alzheimer's disease. And for me, I had already become a neuroscientist, and so I decided, for whatever reason, that this was really interesting and dedicate my life to it. And when I started telling people what I was doing, like when I opened the clinic at Rockefeller down the block in 1981 for Alzheimer's disease and dedicated my lab there to Alzheimer's, people would ask me, what do you do? What do you do? And I say, oh, I take care of people with Alzheimer's disease and I do research on Alzheimer's disease. And they'd say, old-timers disease? I never heard of that. You know, and so we came from an era where that was even before beta amyloid was discovered. And just to fast forward, I mean, what I'm most excited about is, in my professional lifetime, the progress we've made. It's just amazing because, you know, we really knew literally nothing about the illness 40 years ago, 42 years ago. And today we have the first disease-modifying drugs on the market. We have brain scans, we have blood tests. And the advances in clinical practice and in trials is just amazing. So I think, you know, it's been written that from the time of a discovery to the time of a therapeutic is somewhere on the order of about 35 years. There was a research on this, and the illustration is cholesterol was discovered as a risk factor for heart disease in the 1950s. The first statins, the first drugs for heart disease really didn't come to market till the 1980s. And so in a way, like, we're really on time right now from all of this tens of thousands of research papers that have been published over the 40 years to harvest all that information and turn it into translating it into new drugs. Our strategy being, as you've said, Mark, and all of us, that based in the framework of the biology of aging. So what I'm so excited about is the first disease-modifying drugs. They're looking actually better and better, I think. Talking to people that are bringing these to market, there's interesting aspects to it, but it's just the beginning. And so what I'm excited about is that we've entered into a new era of Alzheimer's disease where we can envision this will be a manageable, treatable, and preventable disease.
Here, and Howard likes to say, it's harvest time, and so many of you, and thank you to the Goods family, have been supporting this, and now it becomes harvest time. If you're 60 and above, you have cognitive issues, the options are coming. If you're 30 and above, there are going to be prescriptions, and if you're younger than that, there's going to be precision medicine for you. Thank you to our fabulous panel today. We're now headed to the third floor for lunch, the President's Room. Yes? Yes, President's Room, third floor.
Thanks, everybody.
Thank you.
Thank you.